So this is the theme of the first breakout session. We'll be discussing the scientific and technology challenges regarding the catalysis of neuroscientific discovery and the development of new therapeutics. And prior to the conference, we did pose the question to you, which I won't repeat, but which you know and which is shown here. We got 65 responses, and based on these, we grouped the responses into themes and into the tables where you'll be sitting. So here are sort of the main themes and main tables where you'll be located. So you'll have some commonality, some predetermined commonality with those amongst you. And then after about a half an hour of discussion, we'll reconvene here and sort of report what was discussed and disseminated or determined at the individual tables. We'd like you to focus the particular reports on the following three questions. What are the scientific and technological challenges in the current state of affairs? What does significant advance look like? Where do we need to go to get to the next level? And what barriers or hurdles do you see in getting there?
Let's start with table number 1, and the topic is targeting therapies with cell type specificity in the human brain.
Okay, so we had a wide-ranging conversation, but a couple of themes. One is the technical challenges, a lot of it involved the fact that we're proposing to try and do these things in humans, and all the tools that are currently available for doing this, these wonderful experiments in non-human models have many challenges because of safety and ethical issues in humans. And so discussion about some sort of concerted effort for validating and demonstrating the safety of gene therapy platforms in humans as part of a long-term strategy to allow us to deliver some of these tools in a cell type specific way. There's a question about alternatives to that, especially in the short term, and the ideas some of them brought out earlier in Garrett's talk about using cell surface markers that would be an effective way of concentrating agents in the presence of certain cells and being very identifying sets of cell surface molecules which would identify specifically particular populations of cells as an intermediate strategy, especially with the idea that we can take advantage of the fact that this can be sort of combinatorial or intersectional, so that there might be multiple delivery of multiple agents to the same cells, so that we have an enhanced level of specificity. And there's discussion about stimulation-based approaches or combinations of stimulation and drug therapy. So, for example, being able to stimulate a certain pathway to activate a small population of neurons simultaneously with giving a drug, which might be selectively active on neurons that were strongly activated during that period, as another way of getting at least partially cell type specific approaches. A lot of discussion about some of the challenges associated with FDA approval of these things, the notion that there have to be sort of standardized ways of mitigating some of the risk, the notion of, for any of these things, being able to turn it off, some kind of kill switch, and a systematic evaluation of the relative safety of different approaches to this. Those were, I don't know if there's any other comments. All right, thanks.
Perfect timing, 2:30.
Group 2, targeting therapies with cell type specificity in the human brain.
Yeah, so we discussed a lot of the same things that Nathan mentioned. I think just to add on to the issues that he described, we discussed some of the challenges associated with defining cell types and establishing the relationship between various features like genetics and connectivity and morphology and function within the brain. And also the challenges associated with understanding the relationship between cell types across species and the uncertainty regarding whether cell types in the mouse actually match up to cell types in humans, or which is the more intriguing possibility that there are specific human cell types that have yet to be discovered. And also, we discussed, again, just focusing on things that Nathan didn't bring up, we discussed the importance of not being neurocentric, and in particular, one of us is studying microglia, and the fact that they can migrate to different areas of the brain means that they could be functional in terms of carriers, but also their importance for maintaining brain homeostasis. Let's see. Am I forgetting any? I'm sure I'm forgetting things, but I think that's probably going to cover. Oh, I know. There was also discussion of different ways of accessing the brain. And in fact, we did discuss the strategy that Nathan talked about in terms of specifically targeting cell surface proteins based on their expression patterns. But we also talked about selective permeability of the blood-brain barrier as a way of therapeutic targeting, in a certain sense as a way to get circuit-level specificity that wouldn't necessarily depend on cell type specificity.
So next, we'll do tables 3, 4, 5, starting with table number 3, and the topics for these tables is developing an integrated understanding of brain disorders from genes to circuits to dysfunction.
Okay, well, we came up with lots of challenges to discuss. So first on the list is really the question of species predictability, whether or not mouse is sufficient or not sufficient to translate to human, or whether you need an intermediate in terms of marmosets or non-human primates, or whether or not the mouse models, which have proven not very translatable, are really just not being investigated with the idea of trying to identify circuit-level endophenotypes and commonalities between diseases rather than a reliance on, I think, behavior. The behavior, as a note, is being quite often too crude to establish, I think, the very complex symptomology of patient diseases. Question in regarding to translating a sort of bottom-up approach from genes up to circuits and dysfunction. The challenge there really is in the genetic heterogeneity of many of diseases which aren't monogenetic, and whether that's a good or realistic starting place given the tractability of modification of some of the identified genes and the scaling of the bottom-up approach that would really be required to test the impact. However, clearly, I think everybody realized that this is a critical component of this, and a real necessity for an unbiased sort of systems approach to understanding disease models as much as possible in terms of various unbiased methods, omics technologies, etc. And try and use that in order to identify predictability of emergent phenotypes between disease states in humans and in models. What else do I have? A note that perhaps it's time in some of the human diseases to recognize that one of the biggest challenges we have is actually understanding the circuitry disease phenotypes in the humans, and this is actually preventing some of this work from moving forward in models in terms of therapeutic development. And that the time is probably quite ripe at the moment to try and reevaluate our current understanding of many of these disease models by focusing on, experimental medicine approaches in patient populations with the improved techniques and technologies now available to get a much greater and higher resolution understanding of the disease.
Great, perfect, thank you.
Table number 4.
So this is a tall order to develop this integrated understanding. But we talked about 3 general topics: what the current state is, and then what are the technological challenges and the cultural challenges to getting to a better understanding, a more integrated understanding. So the current state, the group felt like there's a fragmented and incomplete understanding of particularly complex disorders at a circuit level and functional level. And specifically in complex disorders, it's, as we've mentioned multiple times today, it's not well modeled in animal models, so that is a challenge. As far as technological challenges, there does seem to be just a lack of tools to monitor brain activity at a deep level, a molecular level in humans and across brain regions. So developing tools in that area would be hugely beneficial. And also just a lack of really good computational tools to make sense of some of the data that are being generated and shared to make meaningful deductions on what these data actually are saying. So developing better ways to handle these large data sets and complex data sets would be valuable. In terms of cultural challenges, the group felt like there was just a lack of really good, well-defined cohorts, not just of disease cohorts, but normal, quote-unquote normal cohorts at a molecular, that collect a lot of information at a molecular level, at imaging data at a kind of a large functional level, and broad clinical phenotyping, and having more cohorts both in disease and controls would be valuable and really accelerate this integrated understanding of brain disorders. There's also some cultural challenges of lack of opportunities for multidisciplinary teams to work together across biology, computational science, clinical work, et cetera. So if we could have more opportunities for that, that would be fantastic. And then also one final cultural challenge was just promoting standardization and some development of consensus ways to acquire, collect data and samples, ways to curate data and make it available, call it the same thing, developing some standards around ontology would be very beneficial to this integration.
Great, thank you.
Table number 5.
So our group was really struck as well by the high dimensional nature of this problem. So we began our discussion talking about genetics and the high number of genes, the weak contribution of many of these genes to risk for various psychiatric and neurological diseases, and then the multiplicity of animal models that exist. Like other groups, we also discussed the need for greater collaboration between different people so that people could compare their results across those different kinds of models, and other dimensions as well, such as time. So we talked about the long time scales that would be needed to really understand developmental diseases. So one person in our group made a sort of provocative comment that maybe we should just forget genes entirely in order to reduce the dimensionality of the problem, and we discussed the possibility of focusing our attention on circuits that might be conserved across species. Given that the problem itself is so high dimensional, maybe we ought to really look for circuit-based endophenotypes in this way. We also discussed potentially the need for theory in neuroscience, that success might look like having a really good theory of what these circuits do across species. And as far as barriers, we discussed the incentives that exist in science to do this, funding mechanisms. We also at length discussed some of the limitations of inbred mouse strains and genetic backgrounds as a sort of impediment to this kind of research. And then, as I said, the sort of inherent high dimensionality of the problem.
Great, awesome, thank you.
Next, we'll have tables 6 and 7, and the topics for these tables are next generation technologies for an accurate mechanistic understanding of brain disorders.
So, as a table of technologists, we've decided that the technology is not the limiting factor here. So we talked. Our initial discussion was focused on really what does mechanism mean and what do we mean by brain disorders. And so better defining those things would be of great use to actually, without knowing those things, deploying these technologies is not possible. And related to that, there's this question of what is the actual language we use to articulate dysfunction and disease that neuroscience and psychiatry in particular have perhaps a poor language set to define this. But getting past those questions, we were interested in looking at integration of technologies across spatiotemporal scales and trying to understand what the appropriate scale is to look at and to delineate to minimize the scale of the problem. With specific diseases, you may be interested in different scale levels, whether it be cellular or genetic or even circuit level. So to help define that, having more theory, that theory in particular is lacking here. Theory that is built specifically around models of disease was something that came up earlier in the day, and that'd be relevant here. And in terms of barriers to getting to the next level, aside from these technologies we have bits and pieces of these core technologies, would be just integrating sort of the cultural side of getting experts in each of these tool areas to work together on the same disease and same problem.
Great, thank you.
Table number 7.
Yeah, we started off talking about really what the long-term goal would be, and then we got into more specifics of things that could be done in the short term. The long-term goal would be to get to millisecond resolution, submicrometer resolution, receptor-specific resolution, non-invasively, all in a human being. World peace would also be nice. But in the short term, we feel that a lot of the promise is going to be in combining modes, multimodal techniques, and combining models, whether it's rats, large animal models, human beings. The fact that they all have strengths and weaknesses, things using opto-fMRI, so optogenetics to better understand fMRI, which is more relevant to the human model right now, EEG and TMS, using TMS to selectively inhibit to understand the neural circuit pathways, using endoscopic style optical imaging of the current fields from a clinically relevant DBS electrode in an animal model that can do it. Those are all things that can be done and are going to start to be done, but the biggest thing is there's only certain things we can do in human beings, certain things we can do in mouse models. It's having those people talk. What are the strengths and weaknesses? Making sure to establish the human relevance and get a little bit less of the serial translation where we optimize a drug or device in rats, then we wait. It takes about 10 years. We try it in a human being. We know it's not going to work from the gate, but we expect it to work, and we just waste time. We need to create the structures for technology to do this parallel translation where people who are traditionally siloed are always talking in real time about their data to establish the animal model relevance and improve upon it. We talked a little bit about we're going to be getting such data right now, we're going to run into the old drug screening problems where there's going to be a lot of false positives. We're going to be getting human data which has, in an individual patient, a lot of information, but not a lot of N. And so we're going to have to figure out how to deal with these large amounts of data and the false positive rate that we're going to get, and how do we create structures to prospectively screen something that looks like a promising target. And right now we're still doing a lot of retrospective analysis and assuming that everything we see is real instead of a statistical aberration. We also stressed if we're going to study the brain, you really do have to be able to measure and perturb the inputs and outputs to and from the brain, and that a lot of the Brain Initiative talks about this great technology to measure things in the brain, but if you can't selectively perturb the inputs to the brain and measure the outputs, you can't possibly understand the brain. And we need to be able to do that not just in animal models, but to some extent in human beings. We talked a lot about there's more and more of a role for again, to amplify what has already been said, non-neuronal cells, receptor expression, plasticity, neurochemicals as a function of glia and astrocytes, especially for DBS. There's more and more indications that stimulating astrocytes causes neurochemical release that affects all these circuit behaviors. We need to be able to measure things that change receptor expression and microRNA expression, both consistently in an individual animal over the course of weeks to understand those changes over time, but we need to create instantaneous biomarkers to say if this may affect receptor expression over time for something, again, like DBS, where a lot of Helen Mayberg's great work is showing that maybe these patients aren't getting an effect for 17 days. We still need to be able to titrate these patients with second resolution. So we need to find biomarkers that predict the plastic changes before they have the behavioral effect in real time. I think that covers just about everything.
Yes, thank you.
Next is tables 8 and 9, and the topic is biomarker development for better disease characterization and early detection and for objective therapeutic evaluation, starting with table 8.
All right, so in terms of biomarkers, in terms of the current state of affairs, there's kind of a bit of a rift in terms of how advanced some areas are and others are not. We think for psychiatric disease there are, somewhat imaging biomarkers to suggest. We think that it's probably further along in terms of the multimodal approaches for something like the neurodegenerative diseases like AD, where there's PET imaging for A beta tau, metabolic disturbances, tau and A beta in the CSF blood biomarkers. All this is good and well, but it's still not working in terms of identifying patients early enough that the trials are successful. So we think that a lot of the trials are falling apart, partly because we're targeting patients or treating them much too late. There was some discussion in terms of state of the current affairs with genetics, in terms of taking diseases which could, in terms of symptoms, be quite similar but genetically be quite distinct, and that might also be a matter of defining the heterogeneity better. But if we focus a bit more on just the actual biomarkers, the idea would be that we could diagnose patients, substratify them both in terms of disease categorization as well as response to treatment, as well as prognostic ones to track the progression. And ultimately, one of the challenges here is defining that cutoff. So we have something like the natural course of aging, where you have cognitive decline, you have MCI, mild cognitive impairment, and then you have a track to AD. At the earlier stages, when we're looking at the prodromal early detection part, it would be hard to exactly identify one from the other because they're probably tracking similarly and then they break apart. So it's that early phase that we wanted to focus on in terms of what could we do to identify those patients at a population level so that we can intervene early enough. So in terms of and then one kind of side note that we discussed was another way that we could use genetics to inform this would be to use something like basket trials in cancer, where we're not defining it based on the actual pathology, but by the genetic markers that link the disease and might identify susceptible patients. So in terms of advances, if we're going to focus on early detection in prodromal phase, one thing that we think could be harnessed in terms of a 10 to 20 year outlook are the life studies that are being done. Now, these are being done in large cohorts of healthy patients or healthy individuals that ultimately will develop various diseases, and they're being tracked in every possible way, and they're wearing wearable devices to look at their sleeping, their cognitive things, how much they move, their diet. So there's so much information being gathered at a large scale, and perhaps we can harness all of this information being pulled out from these wearable devices and other types of larger information and identify susceptible patients, because we'd love to pop them all in an MRI, but that's not tractable nor feasible. So the idea is to identify patients early enough using fairly non-invasive population-based measures, and then be able to pull them into the clinic and identify you are the person that we need to put in the MRI and look for the clinical correlates, and then enroll you in the trials. And the idea here is to broaden the definition of a biomarker and to integrate multiple types of information so that we can not just have to focus on one thing or the other and not be too reductionist. And then in terms of what is this going to look like clinically, I will hand off the microphone.
So in that perfect world where we have world peace and technology is not the thing holding us back, how much does early detection turn into preventative medicine, and how would we affect that behavior change in a population where if we know the biomarkers and we have the technology early enough to understand what a disease trajectory versus a normal healthy or an average healthy trajectory is, how can we make that a part of treatment and medicine, and can we use a potentially like a vaccine style approach for behavior change in really being that far ahead outside of any clinical or behavioral expression of these diseases and treat that far in advance.
Great, thanks.
Table number 9.
So we actually came up with several very similar themes, although also some new ideas. So biomarkers clearly are going to be critical, and we talked about several types of biomarkers, predictive, diagnostic. Stratification is key because so many of the conditions like autism are so heterogeneous that if we can't identify someone that would be appropriate for a clinical trial, we have no hope. Also as early efficacy indicators, and then improved readouts in clinical trials. One of the things that we talked about that I think is important is actually improving the way that we measure behavior. And so thinking about behavior as a biomarker, and that one of the ways to do that is through developing digital phenotypes. And allowing people to wear sensors or have other sort of aspects of digital readouts that are more objective, more quantitative, and that this will provide a much better readout in clinical trials than we've had previously, which often rely on parent or patient report. One, I think, interesting idea is the idea that we need to actually start with a biomarker to define the group rather than the diagnosis. And I think a quote, and since this came from the former director of NIMH, it's interesting, is the utter failure to develop biomarkers that map onto diagnostic categories. So the idea there is that we're actually hurting ourselves by thinking always in terms of diagnosis, and that we may need to start with the biomarker. It could be a circuit biomarker, it could be another biomarker, and then work backwards from there. An example is, of course, the RDoC project at NIMH. So it kind of elaborating on that idea, that then precision medicine it becomes precision based on that biomarker. And we also talked about the idea of a biomarker profile, so that you're not just talking about one type, but it could be a combination of a digital phenotype, EEG, blood-based, and that it may be that combination that is the most sensitive and specific. We agreed that absolutely the greatest need is for biomarkers that identify people before the syndrome is present, and that this is going to require developing biological signatures or biomarker profiles that have a high level of reliability. And the only way for that to occur, or one way, is to have large-scale prospective studies that allow us to really look at these biomarkers and then look at their predictive validity. And then finally, we brought in the idea of thinking about the role of experience or environment. And so we know that the risk for syndromes emerging when they're detected before the manifestation of the syndrome is present can be altered, but usually there's a sensitive period where experience plays a role, keeping in mind that experience actually shapes those neurocircuits. And so thinking about sensitive periods and the role of stimulation and experience in as interacting and shaping biomarkers will also be important.
Great. So now we'll have tables 10 and 11, facilitating bidirectional research / making progress in the absence of predictive animal models.
Okay, so ditto. No, much of what we came up with are things that I've heard, and just to either reinforce a few points and add just a few things maybe that didn't get highlighted as much. So we believe that, you know, for these complex brain disorders that we really do need a multi-pronged approach. But the question is, what aren't we doing now? So one of the things that we're really not doing much of is this idea of this iterative translational research, where you're kind of going back and forth rather than sort of one-shot clinical trials. So a way to be kind of learning as you're doing these clinical trials and going back to animal models to answer questions and then tweak the clinical trial and keep going forward. So that's one suggestion. With that goes the idea that more interaction between preclinical and clinical researchers would be helpful. A second point is that what we're asking of the animal models is maybe the wrong questions, that we need sort of a smarter, more sophisticated ways to use the models. We need to make the models more precise. So as we have a better understanding of the human condition and stratify them into these subtypes, if we could have models that reflected that, that would be a step forward. And I think that's really kind of the key points.
Great, thank you.
Great. Last group, table 11.
So we had a very wide-ranging discussion, and I think there's very little that I have written down here that hasn't already been said. One of the, maybe two quick ideas. One is that in the absence of predictive animal models, we should be a little more creative. I think this gets back to something that Corey Bargman said this morning, get a little more creative and flexible in terms of thinking about animal models and looking across different animal models. We have Rich Mooney in the group who was talking about some of the successes for looking at things like song production and songbirds, where there can be very quantitative analysis of behavior and linking back to neural activity. We, along similar lines, noted that there's probably much more to be mined from extremely careful observation of patients with disorders that haven't been fully mined, and following very aggressively on some of the low-hanging fruit leads, such as the evidence of ketamine or sleep deprivation in depression, and pushing really hard on some of those interesting findings. Otherwise, I think pretty much everything else was covered.